It is now my pleasure to welcome today's speaker to share his expertise and knowledge about advancements in PH research. Dr. Todd Bull is Professor of Medicine, Pulmonary Sciences and Critical Care at the University of Colorado.
So what are we going to talk about today? Today I've been given an opportunity to discuss really an essential aspect of the care of patients, which is drug discovery and development. And while it's essential to patients, it is, we'll talk about, kind of very early and removed in its infancy, if you will, from reaching the patient. We want sort of to give an understanding or give people an understanding of really what this looks like. So just some terms to start with. What is drug discovery and development? Well, as the kind of the opening sentence begins, it's the process of identifying and then testing possible new medications, new drugs, and then bringing them. The term that we often use is from the bench to the bedside, or more specifically from the laboratory, from the research laboratory to the patient, if you will, from bench to bedside. If you could advance that to the next slide there for Jill. So these drugs are identified through understanding of disease pathways and new technologies, and so this is how we get to this point. And of course, the stakeholders in this scenario are the scientists, physicians, and physician scientists who sort of launch into this research and really focus their careers around it, and then their goals to bring it to patients. Pharmaceutical industries, of which we heard from two who are sponsoring this program today. The FDA works best, of course, when left alone politically and allowed to do what it does. For now, decades here in this country, and does it very well. And then, of course, the most important stakeholder is the patient along these lines. If we could jump to the next slide there. And so this is now essentially an overview, a 40,000-foot view of what it really means to bring a medication to the patient population. And so this starts with drug discovery, and this starts, we're giving numbers here. These numbers are not necessarily specific, but they're examples. So if we started with, for example, 10,000 or more compounds, and that's a realistic number of what we might be considering for medications, then of those, and through a sort of iterative process that moves to the preclinical phase, paring it down as an example to around 250 compounds that might be considered out of that initial 10,000 to be good candidates for preclinical studies. Then to the clinical phase, so now moving this now to patients, that might be then five compounds out of those 10,000, and then finally ending up with one, which is drug to patient. And so so this is certainly a complicated process. And I think maybe it's sort of lost impressive to, or maybe not well understood to hear that how long and how expensive this is. So, going from those initial compounds to final drug is about a 14-year journey. It's about a $2.5 billion adventure. My daughters are homeschooling at the moment, not homeschooling, video schooling. And so one of my daughter's roles in science today was to try to figure out what a billion really meant. And we hear these terms, $1 billion. Anyway, I digress. But this was just an impressive dollar amount to get to one drug, if you will. And then, our goal, of course, is to take these compounds, bring them as quickly as we can and as safely as we can to the individuals. But it's this kind of complicated process that takes so long and costs so much. So today, I'm going to advance the slide here. We're going to be talking really more about drug discovery and that preclinical phase. You can go to the online portion of the PHA, and there's already been discussions about the clinical and the FDA review. So if you didn't have an opportunity to see those, these are recorded episodes you can go back and take a look at. But today we're going to focus on this first part, which is, again, drug discovery. How do we identify potential compounds? And then preclinical. How do we start moving them towards the clinical phase? So let's continue from this point. So how do we get from lab to clinical candidate to discovery here at this point? So we advance the slide here because this is really kind of the sort of the crux of this. Really, as this implies, so drug discovery begins sort of with basic concepts from the laboratory standpoint. Researchers take sort of what we know or what we have learned, you know, sort of the standing on the shoulders of giants, if you will. What has built us up to this point? Where from a research lab can we figure out what we've uncovered from pulmonary hypertension research work, and then what can we now kind of look forward to bring it to understand these pathways, these existing pathways, existing therapies then inform us, and then we can try to pull in new technologies. We'll talk a little bit about those later on that help us moving drug discovery forward a little bit more quickly. Move to the next slide. Many of these new discoveries give us through a high throughput approach to this. These are really sort of genomics, proteomics approach to drug discovery. It used to be we had to do this in a much more, again, iterative fashion, have a hypothesis, search for a gene or a protein, and then try to alter it. These high throughput technologies let us look at multiple pathways at the same time. And then lead compounds that sort of light up in these approaches, can then be looked at to see how they might interact with this particular pathway, receptor, protein, etc., and how they could be tweaked or adjusted to do so more effectively. So this is a fairly famous slide done in a variety of different ways. This is really trying to get at what is the underlying pathophysiology, pathobiology, if you will, of pulmonary arterial hypertension. So WHO Group 1 pulmonary arterial hypertension. And what we're seeing here in sort of panel A, sort of to the far left of your screen as you're looking at it, is what a healthy pulmonary artery would look like, cut in section here. So that's sort of the half of it cut through. See it down below on end. That blue arrow indicating blood, or the direction blood would move through the pulmonary artery. And then as this disease progresses, this is really, again, a disease of the very small, what we refer to pre-capillary pulmonary arteries. So the arteries right before they leave the part of the lung or hit the part of the lung where gas exchange occurs, we see a proliferation of cells. And that's really what this slide is trying to show, is this sort of thickening, increased cells that are produced within the lumen itself. Then as we move to part C, and then eventually to part D, you can see where this vessel wall becomes very narrowed, essentially obstructed, and the possibility of new clot or clot forming because of the slow blood flow is depicted here. And I think that the point we want to get at is that at the end of the day, what we understand about the pathobiology of this disease is that it is a proliferative process, meaning a growth of cells. One of my early mentors in the lab really sort of described this as almost a cancer-like proliferation of cells, cancer being an abnormal cell growth. This is not necessarily with, for example, metastasis, but really kind of getting at that this is the underlying pathophysiology, pathobiology. And once you've blocked up enough of these very small vessels in the lung, we start seeing the pressure rise. If you block up more, we start seeing the heart begin to fail because it's having to push against that hard pressure. And this is essentially what we're now, as a 40,000 foot view again, targeting. How do we figure out how to deal with this particular process? Let me advance the slide here, please. So what do we understand about this, and how has that helped us with drug discovery, drug progression? And so lots of pathways, lots of directions move. So here we see the prostacyclin pathway is involved with this, the nitric oxide pathway, the endothelin pathway, and those are the pathways that we've taken advantage of, but there's many other pathways that we're working on. So here we see angiogenesis as a possibility, other growth factors improving or increasing cell growth and proliferation. Inflammation is recognized to be an incredibly important part of this. Neurohormonal activation, it plays a role. BMPR2 signaling, cellular metabolism listed here, extracellular matrix production. There's BMPR2 signaling, and then calcium signaling. And these are, again, I think a nice summary of what we generally now understand as being important of the pathophysiology. Does not mean that this is the complete list, because a lot of what we're doing going back, is from the laboratory standpoint, is coming up with new hypotheses, exploring new mechanisms for disease. We can move to the next slide here. But these are the ones that we have so far successfully targeted for therapy, and it involves these three pathways: the prostacyclin pathway, the endothelin pathway, and the nitric oxide pathway. And all of our current drugs are focused on these, and we came to these by understanding the pathophysiology, pathobiology of disease. But I will point out, and I think this is an important point to make, that these three pathways predominantly work by altering vasodilatation or vasoconstriction. That is either trying to relax the vessel to let that blue line, that blood flow more easily, or preventing it from vasoconstricting. But I just sort of got through saying that this was a very proliferative process. And while these pathways may have some role in proliferation, really I think our future and our excitement, my great excitement in this field, is actually now starting to hone in on these other pathways that we've listed. So we go to the next slide here, Jim. And so this is what this is stating. So these three pathways that we currently, sort of leverage to, or have leveraged to derive treatment. This is the nitric oxide pathway. Nitric oxide is a very potent endogenous vasodilator, meaning it is how your lungs, when they function normally, open up vessels. So if you need blood to move, more easily to one part of your lung, you upregulate nitric oxide, you let blood flow there. And so we've taken advantage of that. Next pathway is the prostacyclin pathway, again, a potent pulmonary artery vasodilator. Again, this is a way that we can use the endogenous pathway by increasing prostacyclin to improve flow. And then endothelin is a potent vasoconstrictor. It's again a way that you regulate your blood flow through your lungs to do something called ventilation perfusion matching. And so we can block that pathway to help with vasodilatation. And so these are our potential options along those lines. This is, again, just the drugs that we've come up with that are useful in terms of adjusting these. So the phosphodiesterase type 5 inhibitors, the soluble guanylate cyclase agonists, these are the way we modulate nitric oxide. The prostacyclin pathway includes the prostacyclin analogs, and the endothelin pathway alters this pathway, the endothelin receptor antagonist. And again, it's understanding this pathophysiology, pathobiology that allows us to derive these medications. And one thing that'll come up is that frequently these drugs were repurposed for other reasons as we understood this disease state. I think many recognize that the phosphodiesterase inhibitors, that being specifically sildenafil and tadalafil, had another purpose before they were used for pulmonary hypertension. And then it was understanding what was happening in the pulmonary hypertension as well that said, perhaps we could take these drugs and see how they worked in this scenario, and they work well. So next slide, please. So our approaches to pH research for drug discovery, these include really digging in to tissue samples, and this is one of the better ways we can get at this. If we can get samples from human samples that both represent pulmonary hypertension, this being like from explants for surgery or controlled tissues from biopsy, and then culture these out to give opportunities, and then taking those tissues and then using them through a variety of types, such as cellular molecular analysis, proteomic analysis, high throughput genetic studies, histological phenotypings. These are our ways that we can help this in terms of moving this needle forward for discovery. And again, how do we get at this? So we see a pulmonary artery cut lengthwise. We look at the different cell types parsed out here, endothelial cells from inside the vessel, smooth muscle cells from its surrounding, fibroblasts, again, surrounding further, and then pericytes even further out. Then we can take these forward, study these cell types in a variety of systems, trying to figure out what is going on from a cell growth standpoint, inflammation standpoint, migration. Again, this is really, again, digging into this from the science to then sort of inform or give us ideas about what might work from a therapeutic aim. And then, of course, there's the animal models or animal approaches to this. These, we've got a variety of animal models for pulmonary hypertension, ranging from rats to mice to cows and sheep to chickens, monkeys. They're listed through here. Now, what I would say about this is that while perhaps some people are rats, rats are not people. And so while we use these models to try to inform us, and they can help us with certain aspects of the disease, we've been able to cure pulmonary hypertension in a variety of models for some time, and of course not been able to translate that necessarily directly to the human. So they are useful in some aspects of the disease, they are not perfect markers of the disease, hence why we generally also need human tissue to look at this. Next slide. And then we see, as we sort of work through this, we have an idea or a preclinical drug that we think might be useful. We can take this now to various models, try out, see if we improve pressure, if we improve vasodilatation, if we improve flow, and then often we have to do, to do this, we have to confirm this in multiple animal models. It's kind of the norm now to not just accept one model because they all have their limitations when we're looking at drugs, but try to inform us on multiple models. Next slide. So preclinical research, does it work? Is it safe? This is, again, essential aspect of it. We need to make sure that what we're bringing to people, A, has a reasonable chance of being effective. Because you're asking someone to participate in a clinical study, there needs to be some, a good chance that what you're testing is going to be beneficial. I mean, we don't know for sure. If we knew for sure, we wouldn't test it, but it's important that we have a good feel for it. And then most importantly, that it's safe, that we're not putting people at increased risk. So we're looking at our benefits, potential benefits of mechanisms, potential adverse effects, side effects from that standpoint, understand on how to administer the drug, the dosing of it, how it can, drug can be absorbed, metabolized, and distributed, and then early indications of effects on specific patient populations.